Good day, everyone. We're reviewing Wetzel's Limnology textbook, edition number 4, and we are in chapter 11, oxygen. And more specifically, right now, we'll be looking at the spatial variation river network scales. In contrast, larger rivers draining lowlands show lower and more spatially variable oxygen concentration than smaller streams. Factors that control oxygen concentration, such as organic matter supply, temperature, and vertical stratification, are more spatially variable in low-gradient systems, draining urbanized or agricultural catchments. In addition, increased geomorphological and habitat complexity in unmodified rivers is linked with enhanced activity and diversity of metabolic pathways, and thus larger oxygen variability. For example, in backwater areas of large rivers, water residence times are longer, limiting the advection of oxygen and promoting marked variation in oxygen content due to photosynthesis and respiration. Reduced turbulence and advection in low-gradient rivers limits the replenishment of oxygen-depleted waters during downstream transport, leading to a progressive reduction in dissolved oxygen. Oxygen consumption from chemical reactions with dissolved organic and inorganic reduced compounds can be large. In very productive wetland floodplains, as in the Pantanal, very productive Pantanal wetlands of the Paraguay River in Brazil, high rates of organic matter decomposition can lead to high bacterial production of methane. Subsequent bacterial methane oxidation can deplete dissolved oxygen in backwater areas. Moreover, lowland rivers are more prone to be in urban and agricultural watersheds. Rivers in human-modified watersheds are often subsidized with high organic matter and nutrient loading. Riverine eutrophication can result in biochemical oxygen demand that exceeds physical reoxygenation and autotrophic production. River oxygen depletion can also be a result of hydrologic alteration associated with damming or water extraction. Hydroelectric dams that release deep water from reservoirs often create hypoxic conditions downstream due to both low oxygen and high nutrient concentrations. In addition, the presence of large dams or withdrawal of water for irrigation can cause the lantification, when water stops flowing, of running waters, leading to reduced atmospheric exchange rates and lower oxygen supply in downstream reaches. The lotic-lantic interplay can occur naturally or in human-modified river networks and determines the extent of well-mixed versus more spatially and temporally heterogeneous river flows. Consequently, these dynamics are also a key factor in predicting dissolved oxygen dynamics in river networks. Reach and sub-reach scale. The hydraulic connections between surface water and groundwater are a major driver of spatial variability in oxygen concentration, both when groundwater flows into a river or when surface water flows downward to the groundwater. Groundwater becomes naturally depleted in oxygen due to lack of recharge, aerobic respiration, and chemical weathering. For example, in alluvial aquifer river system, flow zones often determine the location of seasonally variable low oxygen sites within river reaches. However, as oxygen-depleted water reaches the river channel, surface reaeration occurs. Conversely, losing oxygen do not normally imply a net gain or loss of oxygen with respect to surface water. At a smaller spatial scale, the transfer of oxygen can also occur between surface water and the riverbed due to the presence of logs and sandbars and the sequence of alternating topographic highs. In such cases, water infiltrates the subsurface and returns to the surface after spending some time in the hyporeic zone. With surface waters often near oxygen saturation and the subsurface often oxygen limited, the flux of oxygen from the surface to the hyporeic zone dictates the magnitude of oxidative processes. Thus, oxygen consumption rates are determined by the hyporeaic exchange rate and the biological activity of the microbial communities in the hyporeaic zone. Overall, the interplay between physical transport processes and microbially mediated biogeochemical reactions generate high spatial variability in oxygen at the subreach scale. So let's look at these figures here. Figure 11-6. So the spatial distribution of dissolved oxygen concentration varies over time, with a shallow alluvial aquifer floodplain river system. So this is A, right here, those two. So we're dealing with, once again, the shallow alluvial aquifer floodplain river system, and B, hyporeaic zone of a riffle pool sequence. River flows from right to left, so it goes from here to there. So this is high in topography, and this is lower in topography. Upper panels show the location of sampling sites, including piezometers and shallow riparian monitoring wells. So this is the same than this, but however, the same figure here. However, they removed distribution of the dissolved oxygen concentration, which is in red and blue here. And then they showed to us the location of the piezometers. And piezometers are shallow well apparatuses where it's pretty much like a pipe that is lowered into the ground and where you can sample water from. And it's not a very large pipe. It's usually about a couple inches in diameter. And so the Nyack floodplain of the Middle Fork Flathead River located in northwest Montana. So this is in northwest Montana with the Middle Fork Flathead River. So the river is right here, I believe. And then B is the Truckee River Riffle Pool Study Area located in northwest Nevada. So this is the Truckee River. And I'm not sure where it is exactly on this map, but that's what it is about. So what can we glean from here? We have the scale here, dissolved oxygen in milligrams per liter. So when it's yellow, we don't have very much oxygen, and as we saw before, we have close to hypoxic conditions when it's hovering in anoxic condition when it's less than 1 milligrams per liter. And so in blue, it's higher. And here we have kind of the same system, but it's red is 5 milligrams per liter and blue is 0.1 milligrams per liter. So the first thing that you can see is there's a lot of variability in the oxygen concentration in the water of those rivers. I mean, it's extremely spatially variable. The other thing that you can see is that through a year here we have, so here we have October, and then this is November, October 2008, and November 2009. Here it's in January through October. I mean, yeah, exactly. And so January, April, July, and October. So at in the winter, that river has higher oxygen than it does in October. And so what probably happens is that during the summer months, you have a growth phase that is utilizing oxygen concentration, such as vertebrates, invertebrates, and I guess to some extent plants, if there is some due to the presence of respiration. This is figure 11-7. Hypothetical trends of dissolved oxygen concentration and nitrate concentration and nitrification NP, denatrification DN, and methanogenesis along a hyporeaic flow path within a gravel bar. So here, the first thing that we looking at is the fact that it's not data that was collected. This is just based on a model. This is based on our understanding of how oxygen, nitrate, dissolved oxygen, denatrification, and then methanogenesis would occur. So on and this would be like in the hyporeaic zone of a gravel bar. Okay, so the y-axis here is the oxygen nitrate concentration, and then here is nitrification NP, denatrification DN, and then methanogenesis MP. So we have MP and NP. N as in November, M as in Mike. So this is a gravel bar, and so the flow through the gravel bar. So we are looking at these concentration in the gravel bar as we move with the water in the gravel bar. Okay, so first thing is oxygen concentration is pretty high. NP, which is nitrification, is pretty high as well. However, the nitrate has not yet seen high, so there's a lag. So there's nitrification, and then it's pretty high and decreases. And we have an increase in nitrate here. Then we have DN, which is denitrification. So one is the opposite of the other. This one removes nitrogen, and this one adds nitrogen. So adding nitrogen creates nitrate. Like removing nitrogen through denitrification decreases the nitrate concentration. And then it's getting replaced by methanogenesis, so with the production of methane. So very likely if we had another curve here, we would go down like this. We would have oxygen concentration at its high here, and then it would decrease, and then you would have, instead of nitrate, you would have methane concentration here. All right. The length of aporeic flow path determines the residence time of water, influences the biogeochemical processes within a river ridge. For example, a suite of redox reaction may occur as surface water moves through the sediment of a bar or riffle, so that the concentration of electron donors and acceptors, particularly of dissolved oxygen, would vary following a predictable thermodynamic sequence. First, dissolved oxygen is consumed by aerobic respiration, and next, nitrification of ammonium produced by the mineralization of organic nitrogen increase nitrate concentration along the hyporeic path. As dissolved oxygen is depleted, denitrification may remove nitrate produced at the upstream end of the flow path. Consequently, aporeic flow systems within short sandbars are normally well oxygenated and are a source of nitrate to the river, whereas longer bars would be a sink of nitrate caused by the increased residence time and low oxygen subsurface water. Temporal variation. Rivers exhibit a broad range of dissolved oxygen fluctuation associated with processes operating on temporal scales of seconds to decades. Each temporal scale of oxygen fluctuation provides different insights into the structure and function of rivers. Below, we discuss temporal variability in oxygen dynamics at different scales of ecological and biogeochemical relevance for river ecosystems. This includes the signals of processes operating over fractions of seconds, from seconds to days, from days to weeks, or even several months, and longer term patterns, including annual to decadal variability. So before we move on, let's look at this figure here, figure 11-8. Time series of dissolved oxygen at the surface of the Cinca River in Spain, illustrating how observed pattern can vary a decadal. So this is decade. So this is not model. This is actually, data that was collected in the field. B, we have multi-annual. So this is decadal. This is multi-annual. C, seasonal. D, episodic. So sometimes, like specific events. And E, diel scale. So basically daily. So here is on the y-axis we have the dissolved oxygen concentration in milligrams per liter, and then here we have the time. So the time can be years or it can be days. So what we see first is variability, right? So we have like we have how we're pretty actually we have extremely variable concentration of dissolved oxygen before the 2000 in that river, in that Cinca River in Spain. And then it somewhat stabilized, and then it appears that there's a slight increase through time of the dissolved oxygen concentration from a decadal perspective. This is between years, so that probably is between 2007 and 11, so it's about 4 years. And so it's repeating basically the pattern that we're seeing here. Now we are seeing it at a finer scale, because for example, like the 2009 time period is right around here. And so this is this one right here, okay? Then dissolved oxygen is seasonal. This is based on the season. So we just saw like in the winter we have higher dissolved oxygen concentration in the water than we do in the summer. So this follows this pattern. This is episodic. This was not. This was collected from between March and April over like a month period, so following the same pattern, and then time. And then days of the weeks. And so, in this case, we can see like that there is overall the trend of concentration in the concentration of oxygen is actually decreasing for those like at that scale. All right. So let's see annual patterns. The relative importance of the physical, chemical, and biological drivers of dissolved oxygen content in rivers changes throughout the year. These interacting processes determine a river's annual oxygen pattern. Annual oxygen patterns are mostly driven by the seasonal fluctuation of photosynthesis and respiration, dependent on flow, light, and temperature conditions. Below, we describe different examples of river annual oxygen dynamics classified according to their degree of seasonality and address potential controls. Annual patterns in seasonal rivers. The highly productive Menominee River, a large river in northern Wisconsin, shows a regular and predictable annual pattern with large summer peaks and constant low oxygen under ice, buried under ice during winter. This river is an example of how both the low hydrological disturbance and unlimited light availability drive summer productivity peaks as depicted by large daily oxygen swings. As fall turns into winter, rivers in snow-dominated regions normally enter a dormant phase in which biophysical processes have a minimal effect on oxygen saturation. However, oxidative processes at the sediment in large rivers can promote some depletion of oxygen throughout the winter, although at a lower rate than in the summer because of colder temperatures. In small rivers, ice formation during winter interrupts the exchange of oxygen with the atmosphere, and groundwater chemistry can impose a constant baseline of oxygen saturation. As winter turns into spring, elevated discharge sustained by snowmelt increases light attenuation and physical disturbance, which are both unfavorable for oxygen production by benthic algae. At the same time, increasing discharge also increases turbulence and hence atmospheric exchange, keeping oxygen near saturation. The Famo Creek, a easily shaded and frequently heavily shaded and frequently flooded small river in western Oregon, shows that the phenology of terrestrial vegetation leads to transition in the light regime during the leaf out and later fall periods that can be particularly pronounced in temperate streams. Although it is well known that light regimes primarily drives riverine productivity variation over time, regular and predictable littoral patterns in the fall fuel high in-stream oxygen demand, which results in persistent low oxygen conditions. Dissolved organic matter leached from soil following precipitation events or loaded from organic waste, sewage sources can similarly increase microbially mediated oxygen consumption of oxygen at a shorter time scales. So let's look at figure 11-9 here. So temporal trend in dissolved oxygen percent saturation for 4 contrasting rivers over a year period. The Menominee River in Wisconsin, this is A. B is Fano Creek. C is 5 Mile Creek. And then D is San Antonio River. Here we have, on the y-axis, we have the concentration of dissolved oxygen, basically, dissolved oxygen concentration. And then on the x-axis, we have the years. And so what we can see is there is a very high variability in the Menominee River compared to the others. There's not as much variability in terms of swings in the San Antonio River. And then the 5 Mile Creek definitely kind of somewhat sits in between. The lowest where there's actually really not as much variability, at least from a diel perspective, from a daily perspective, is the Fano Creek River in Oregon. And then we have this sort of long, drawn-out pattern here with very low concentration in that creek, and probably because, as the text was saying, this occurs during the summer when the summer productivity is, because that's a high demand of oxygen. And then here in 5 Mile Creek in Alabama, what is interesting about it is we don't really have those periods here, like where we have low oxygen concentration. I mean, we do have some here, that's somewhat of an anomaly, I guess. But otherwise, we are dealing with pretty good swing of dissolved oxygen concentration on a daily basis for that creek. And I think that's going to be it for today, and I'll see you tomorrow, and we'll pick it up from, I think it's going to be in nutrient limited oligotrophic rivers. Thank you for your attention, and I'll see you tomorrow.